So, reproduction. What is reproduction? Ability to produce its own species. So, ability to perpetuate its own species. Perpetuate means multiply its own species. What are the types of reproduction? Asexual reproduction, or is called vegetative reproduction. Another one, sexual reproduction. What is asexual reproduction? Reproduction without the involvement of gametes. Reproduction without the involvement of gametes is called asexual reproduction. So in asexual reproduction, lower group of plants reproduce by fragmentation method, budding, spore formation, gemma formation, etc. Binary division. These are lower group of plants. Whereas in higher group of plants, vegetative reproduction involves buds. Buds where they develop? Buds develop on the stem, buds develop on the root, buds develop from the leaf. These buds develop into new plants when they get detached and planted in the soil or under suitable environmental conditions. They grow into new plants. Now, already I have seen about buds in the roots. What kind of buds in the root? Adventitious buds. Buds develop from plants other than a terminal bud, axillary bud. Such buds are called adventitious buds. Adventitious buds on the roots develop into new plants. So few plants you have seen that Ipomia, patatas. Then Ipomia patatas. One example, Ipomia patatas, buds on the roots. Moringa, Psydium guajava. So in all these plants, moringa, moraya, etc. So in such plants, buds develop from the roots. They produce new plants when they get detached and grow on the soil. Then next one about buds from the stem. So which plants produce buds on the stem in the sense? All plants produce, but typically underground stem and runners. Of course, subaerial stem. Plants with underground stem, subaerial stem, they produce buds. Such buds develop into new plants when they get detached. Already I have seen about that. So what are the four types of underground stem? Rhizome, comb, tuber, bulb. So in all that, buds develop from various parts, they develop into new plants. Of course, such buds are called adventitious buds. So like that subaerial stem, runner, offset, stolon. These are the few examples. Now about the leaf. Buds develop from the leaf, they give rise to new plants when they get detached from the parent plant. So what kind of buds are they? Buds develop from the leaves in the sense, which parts of the leaves give rise to buds in the sense? In bryophyllum. What is bryophyllum? Katti putti podum. In bryophyllum, the leaves are notches. The leaves are notches. Notches means the leaves are thick and succulent. So here you get notches. So the buds and the notches develop into new plants. How they develop into new plants? These are adventitious buds. They produce adventitious roots. And then when get detached, they develop into new plants. What it is? Bryophyllum, Catipota, Putipodum. The next one, begonia. Bryophyllum, begonia, Scilla. In Scilla, the leaves, the foliage leaves are large. The foliage leaves are large, like this. Large, and they produce buds near the tip. So these buds develop into new plants. So this is all about insula. Foliage leaves are long and narrow, develop into develop buds at the tips. Advantages of natural vegetative reproduction. See, advantages of natural vegetative reproduction in the sense, plants produced by this method are genetically identical. So plants can be produced within a large number of plants can be produced within a short period of time. And then horticulturists, farmers use this method for cultivation, harvest plants in large scale. Then the disadvantage. What's the genetic? The only one disadvantage is that genetic variations will not appear. So of course, what's the use of genetic variation? Genetic variation helps the plant to adjust with the changed environmental condition. Of course, that will not develop. That's not one of the drawbacks over there. But what are the advantages of this one? In the sense, plants can reproduce within a short period of time. So now we have just seen about natural method of vegetative propagation. Of course, vegetative propagation by artificial method. Artificial method means so man-made process. That's what actually you have to cut the plant and then plant it in the soil. That kind of method is cutting, layering. So this and all. And then of course micropropagation, tissue culture method. This and all man-made method. So such methods are artificial method of vegetative propagation. So in that artificial method you get two types: conventional method of vegetative propagation, modern method of vegetative propagation. Conventional method of vegetative propagation in the sense, see, conventional commonly followed by peoples for a long period of time in the sense, even from ancient time. So actually our ancestors used this method. That's what conventional method of vegetative propagation. So which are the conventional method of vegetative propagation? Means cutting, layering, grafting, and then you will study about layering. So what is cutting? See, cutting in the sense, best example, sugarcane. The same is there. You have to cut 10 segments are there in the sense, if you cut 10 segments with each segment with the axillary bud, if you plant it in the soil, it will give rise to new plant. So this method is cutting method. So which plants can be propagated by cutting method in the sense, bougainvillea, moringa, hibiscus rosa sinensis, and then next one about root cutting. Root cutting also used for that. Example, malus. Leaf cutting, bryophyllum begonia. Then stem cutting is used widely for propagation. Which propagation? Vegetative method. What kind of propagation is it? It's a conventional method of vegetative propagation. So in that conventional method of vegetative propagation, normally you used to do that. Cut one part of the stem of moringa. Then if you plant it, it will give rise to new plant. Of course, the flower, and then at the same time, if you just plant the seed, but it won't produce plant immediately, it won't give yield immediately, it won't produce flowers and fruits immediately. It will take more time in the sense, it will take 1 year. But at the same time, cutting method in the sense, if you just plant it, it will just produce flowers flowers and fruits within 1 month, within 3 months, 2 months, like that. Okay, so this is what this method is used for, widely for propagation, multiplication. Then grafting. So grafting அப்படினா ஒட்டு போடுதல், grafting. அது வந்து வெட்டுதல் சொல்றது, இது ஒட்டி போடுறது, grafting. So in grafting method, two plants are taken. Actually two plants of same species, of course. Two plants of same species are taken. Two individual plants are taken. Then in that individual plants, they are just joined as one. Two different plants joined as one. So when you are how to join in a sense, so these two plants, in that one part of the plant where the root is used, basal part of the plant is used. Another one, upper part is used. So when root portion is used, that's what called stalk. Then the upper part is called scion. And then now how it will be the character of both. Of course, so this method is used, which plants this method is used in the sense, sapota, A-cross sapota, then mango, citrus, all these plants, this method is followed. Then actually, which part is called stalk? The part of the plant with the root, that's what called stalk. Part of the plant with the upper part, other plant with the upper part only used for the grafting, that's what called scion. Then what are the types of grafting? There are various types of grafting. Bud grafting. See how bud grafting is made in a sense. So in a stem, a T-shaped incision is made. So bud of another plant is placed in that, allowed to grow. That's what called bud grafting. Then next one, approach grafting. This method, stems of equal size are allowed to join together, and then in that one part of the plant, one plant, lower part is used, another plant, upper part is used. So this type of grafting is called approach grafting. Then tongue grafting, in the sense stems of equal size are used, and then they are cut obliquely. The stems are cut obliquely and joined like this. This one is stalk. This one is scion, like that used. Then crown grafting. In crown grafting, the stalk is larger in size, of course, like this, larger in size. Then in that, the upper part, actually scion is inserted into that. So that's what called crown grafting. Wedge grafting. So actually, stalk scion placed here and here like this. Wedge grafting also just similar to this. Of course, here also stalk is large, and then a wedge is placed in that, and then the plant, the scion is placed into it. That's what called veg grafting. So these are various types of grafting. The next one about layering. So what is layering? So the layering in this, no, the stem is bent and it is allowed to soft stem with the plant with soft stem is allowed to bend, and it is allowed to bend into the soil, then placed with the soil. Then after some, the upper part, the leaves are here. Then after some time, what happens, roots develop from this, and then if you cut this part and allow it to germinate in the seed, it will give rise to new plant. So this method of propagation is called layering. So layering is of various types of layering is there. Layering, mound layering. Of course, this method is mound layering. Then next one is yard layering. So yard layering in the sense, actually this is what rooted in the soil, and then here the part is there. Then here we have to apply it with the hormone, clay with the hormone. Now what happens is roots develop in this part, and then it will give rise to new plant when they get detached. So this kind of layering is called yard layering. So these are all conventional method of propagation. Now in the conventional method of propagation, conventional method of propagation? Advantages of conventional method of propagation. So what are the advantages of conventional method of propagation? As I told you already, this method, conventional method, plants can be propagated within a short period of time. Plants can be grown at any time, any place, and then cultivation can be made within actually fruits can be produced, flowers can be produced within a short period of time. So this is the cheaper method to propagate plants. And then the plants can also be grown easily in the places where they cannot grow. So these are the advantages. So disadvantage in the sense, new characters cannot develop. New characters cannot develop in the sense, what if the new characters cannot develop? See, actually the genes of two different plants will not coming together. So that's why new characters will not be developed in this method, vegetative reproduction. Of course, next one about modern method of vegetative propagation. What it is modern method of vegetative propagation? Modern method of vegetative propagation, plant tissue culture method is used. As I told you already, vegetative propagation, one advantage in the sense all plants are morphologically, genetically identical. They can be produced, large number of plants can be produced within a short period of time. And then the plants today produce flowers, fruits so quickly, but you do not wait for a long period of time. So these are the advantages. In the modern method of vegetative propagation, in the sense, plant tissue culture technique used for this. Micropropagation. Already I have studied about micropropagation, plant tissue culture technique. So what is the plant tissue culture technique? The plants, actually cells are isolated from the plant. These cells are allowed to grow artificially in the laboratory under controlled condition. From each and every cell, new plants develop. So that what happens here within the here also, what's the advantage of this? You can just produce large number of plants within a short period of time, and then which can be supplied to the farmers. But of course, more number of plants can be produced, and then plants are alike, genetically alike. Already I have seen about that one in the natural method of vegetative propagation. All plants are alike in the sense They are collectively called clone. They have the same genetic combination, genetic character. So this is what the advantage of this one. Then what is the disadvantage of this one? In all these, the new characters cannot be produced. Then of course, in vegetative method, one plant affected with a disease, all plants would get the same kind of disease. So this is what the disadvantage. But of course, it's a actually costly process. Plant tissue culture method. In plant tissue culture method is a costly method. So you'll have to spend more time for that. You have to spend more money for that. So and then you have to do in the laboratory. Actually nowadays agricultural department, they are just supplying more seedlings of this plant. Then another advantage of this one plant tissue culture is rare plants can be propagated quickly. Rare plant means already you get only one plant, only one tree in the world in the sense, from which now you can just produce more number of plants. So this is what the advantage of plant tissue culture. So now we have seen about vegetative reproduction. One, vegetative reproduction, natural method of vegetative reproduction. Which are the natural method of vegetative reproduction? Asexual reproduction, lower group of plants. Then next one about vegetative reproduction in higher group of plants. Vegetative reproduction by roots, vegetative reproduction by stem, vegetative reproduction by subaerial stem, vegetative reproduction by the leaf. Already I have seen that. This is under vegetative reproduction. This is under which method? In the sense natural vegetative method. Then next one, artificial vegetative propagation method. Artificial vegetative method means that you have various types. So which are the various types of artificial method of vegetative reproduction in the sense? So the first one, cutting, grafting, layering. Cutting, grafting,